Okay, Dr. Palomo, thank you for the correlate. Now, we'll give everybody probably 5 minutes just to prep yourself for another subject. I will start the correlate in biochemistry in 5 minutes. So you take a break for a bit.
Thank you, Doc.
Is everybody here? Can we start now? I can see 51 people, including the one who's doing the correlate. So there are people who are not present, I guess. All right. So we're going to start the correlate now. To start off this morning's correlate in biochemistry, we start with glycoproteins. Wait. All right, as an introduction, glycoproteins are oligosaccharide chains covalently attached to their polypeptide backbones. So they have different functions, and all of these you can read in Harper's. Just be familiar with them. You don't really need to actually memorize each function. And the glycoprotein has an oligosaccharide chain. And this oligosaccharide chain found in the glycoprotein also serves a specific function. And these are the various functions. I think these are more important for you to know than each of the functions of the specific glycoproteins. This is in tabular form, located also in Harper's. Okay. Now, glycoproteins have different functions when it comes to cell interaction. Now, for example, this portion is a specific glycoprotein embedded in your cellular membrane, and the protein portion is seen in green, whereas the oligosaccharide or the glyconutrient is that red thing sticking out into the extracellular space. Now, these serve also as attachment points for certain cells or toxins or viruses. Can you give me an example of a toxin that binds to your oligosaccharide? Vince, Edward. Vince, Edward, are you there?
Yes, Doc. Good morning, Doc.
Morning.
Mga katong come again, Doc. I'm so sorry.
So we just go along here. Toxins, viruses, bacteria.
Yes, Doc.
So what are toxins that combine with or link to your oligosaccharide? There are many. You can mention and then you might just
Like
get it.
mga toxins, Doc, ay usually some of their examples maybe yung katong mga I think mga radicals ni mo, Doc. I think one of them. Yeah, toxins as well. Okay. Mga I'm not so sure, Doc, pero sa viruses kay I think
No, later na.
Okay,
Just a
sige,
toxin. You haven't mentioned any specific toxin. Let's call on Von. Are you here, Von?
Yes, Doc.
Can you give me an example of a toxin which links to your oligosaccharide?
I'm not familiar with any specific toxins, Doc.
Well, just to give you an example, you have your botulinum toxin, you have the Botox. You're familiar with Botox. That links with your oligosaccharide, and even your cholera toxin, which causes, of course, cholera, links to your oligosaccharide or your glycoprotein portion of your glycoprotein. Okay, thank you, Von. What about Joanna or Telano? Acknowledge. Joanna, are you here or are you absent? Joanna is absent. Who is the group mate of Joanna or Telano? RG Grumo from group 4. So I will mark Joanna absent for today.
Morning, Doc.
RG Grumo, is Joanna absent?
Dito siya kanina, Doc.
Okay, so she's absent for my correlate. Tell her. So RG, what are examples of viruses that link with the oligosaccharide portion of your glycoprotein? What are viruses?
Sorry, Doc, again.
Viruses which link. Look at the what do you
Okay.
call this? The slide. What are viruses?
Usually yung mga sample of a virus, Doc, that links to glycoprotein is mga Ebola, Doc.
Ebola.
Mm-hmm.
Okay, yeah. What else?
Virus.
More common than that. Try not to give very rare or very uncommon examples. Very common, influenza, A. You have your HIV. No? Okay, so take note of that. So good job, RG. How about CG? Hi, CG. I saw you kanina. Are you there? Or are you absent?
Hello, morning.
Good morning. What about bacteria, CG? What are bacteria that link with oligosaccharides in glycoproteins? You know, these are all in Harper studies, are not something like wildly from somewhere else.
I'll just guess, Doc, maybe your
TB bacteria.
What is given in Harper, very classic example is your H. pylori, which causes peptic ulcer disease, no? And you also have certain parasites like your Plasmodium, which causes malaria. And incidentally, your Plasmodia have GPI-linked glycoproteins. So take note of that. That's very distinguishing. Thank you, CG. These are the predominant sugars or your branched heteropolymers composed primarily of dehexoses, except, what is the exception? Of course, your dehexoses are your galactose, glucose, mannose. You already know that from the previous unit. And you also have N-acetyluramic acid. You have your fucose. Then you have these other derived monosaccharides like your aminohexyls or your hexosamines and your 5-carbon sugar like your xylose. And these are the nucleotide sugar donors. They're the ones that donate the sugar. They also carry it and donate it. Later on, when we discuss biosynthesis, you will know that these are present. Don't really need to memorize each specific one. Just take note that some of them are important, especially in your synthesis of your glycoprotein. There are 3 major classes of glycoproteins. I'm sure you all know this by heart. You have your O-glycoside link or your O-link. You have N-glycosidic linkage or your N-link. And your very special GPI anchored or your GPI link. Let's play a short clip.
Secretory and membrane proteins are often post-translationally modified with sugar chains called glycans. Glycans are essential for the stability and function of the mature protein. They affect enzymatic activity, half-life, and receptor binding, among other biological processes. Proteins can be modified with different kinds of carbohydrates, N-glycans and O-glycans being the most common types in eukaryotic systems. N-glycans are attached to asparagine residues of the protein, shown with an N. N-glycans have a common biosynthesis pathway, which is reflected in the common structure of their core. N-glycans can be high mannose, which means all the component monosaccharides are mannose residues, with the exception of the 2 core N-acetylglucosamine or GLICNAC residues. N-glycans can also be complex, which means the mannose residues are only present at the core and the branches are extended with other monosaccharides, such as N-acetylglucosamine or GLICNAC, galactose, sialic acid, and fucose. O-glycans start with the addition of an N-acetylgalactosamine, or GalNAc residue, to a serine, shown with an S, or threonine, shown with a T. O-glycans are often short chains consisting of only a few sugars. Or they can be extended by the addition of different monosaccharides, resulting in a longer chain.
All right. So distinguishing characteristics of the N-linked and the O-linked, as you will note later on, because I'm going to keep repeating this again and again. So for the N-linked, which is shown in the figure above, is linked to an asparagine residue, and this is the N-linked portion over here. And then the sugar linkage is your N-acetylglucosamine, or your GlucNAc. Whereas your O-linked has a serine or threonine link. This is the O-link. And then the amino sugar is N-acetylgalactosamine, or your GalNAc. All right, so here, this just shows the figure of glycoproteins which are free and which are membrane bound as well. Now you have O-glycans, I mentioned again, serine and threonine, and your N-glycans linked to your asparagine. Now here is the figure of the GPI-linked glycoprotein found in Harper's. So this is your GPI, by the way, stands for what? Desil Marie. Desil Marie Yanko, hi.
Yes, Doc.
What's GPI?
GPI is what they call this glucosyl phosphatidyl inositol.
Mm-hmm. You sure?
Yeah, I think so.
Okay. You didn't discuss this in your, what do you call that? Your PBL?
Oh,
Because right here is your phosphatidyl inositol portion shown, and this is your oligosaccharide. And in above portion is where your protein attaches. Now the specific proteins will be discussed later on, and they also serve various functions, especially in certain diseases, which we'll discuss later on, involving your red blood cells. Thank you, Denzel. Okay, so we'll start with your O-glycosidic linkage. They have different subclasses. You have your GalNAc and serine or threonine linkage, which is found in mucin. Your gal-gal, sil, serine, found in proteoglycans. Your gal-hydroxylysine, of course, we'll discuss that when we discuss collagen later. And your GalNAc, serine or threonine, found in nuclear or cytosolic proteins. Mucin is a very typical example of a glycoprotein with a very high O-linked oligosaccharide content. More than 50% of this oligosaccharide, of this glycoprotein, is carbohydrates. So they have repeating amino acid sequences, tandem repeats, you call it. And in the center of the polypeptide backbone, you can find them. This mucin is rich in serine, threonine, and proline. And this is the structure, the backbone, and that yellow block shows the tandem repeats. And then these are the oligosaccharide attachments. Now in Harper's, you'll be able to read the function of mucin, physicochemical properties of mucin, and the biomedical importance. Biosynthesis of O-linked glycoproteins. The summary is in tabular form. I think it's in paragraph form in Harper's, but later on we will try to discuss that. But this is just the summary of the main features of the biosynthesis. Okay. Now, I'll just go through them very quickly lang. So built up with a stepwise addition of sugars from a nucleotide sugar donor. The examples were already given earlier. And it involves glycoprotein glycosyl transferase as the enzyme acting in a stepwise manner, meaning, of course, step by step, right? Each transferase is generally specific for a particular type of linkage. And the enzymes involved are located in various subcompartments of the Golgi apparatus. Each glycosylation reaction involves the appropriate nucleotide sugar, of course, as the donor, and O-glycosylation occurs, take note, post-translationally at serine and threonine residues. Okay, so knowing that and looking at this figure, Mark Marius Mopon.
Yes, Doc, good morning.
Good morning. Okay, good morning. What do you think this is? Is this N-link or O-link? So in the figure, you'll have these different nucleotide sugars, the donors. You have these glycosyl transferase enzymes. So what is extruded extracellularly here?
I think it's O-link, Doc.
Why?
I'm actually basing it on the precursor sugars, the galnac and glucnac.
Mm-hmm. And remember, we briefly talked about the major features of O-link biosynthesis in the previous slide. So this is just like typical example of an exam. There will be stuff that can throw you off and make you scared. What are these stuff here? But if you just take note of what we mentioned earlier, just the simple ones, like if, for example, a specific glycosyl transferase enzymes, see? And you mentioned also the donors, that's right. You will automatically know that this is, yes, O-link, so you're right. You can eliminate the rest. The rest are just for distractions, especially during exam. If you know the basic, then you'll be able to answer. Good job. Next is the N-linked. Wait, who is this? Can we admit Leia Inyagrengia? Yeah, I think someone texted her to be present, otherwise she'd be marked absent. So anyway, N-linked glycoprotein, again, has an asparagine glucnac linkage. And the three major classes of your N-linked are your complex, hybrid, and your high mannose. Okay, so do we really want to know the linkage for each? Like from a glucnac in the asparagine in complex N-linked is a beta 1,4. Yeah, let's not do that. Otherwise, make your life miserable. So just focus on what is shown in, this is the old Harper's, this is the new Harper's. So not as complex, not as intimidating, but just take note of the terminal oligosaccharide, which is the distinguishing part of the oligosaccharide. So for your complex, you have sialic acid, galactose, and glucnac. Whereas in your hybrid, you can find both gal, glucnac, mannose, and in high mannose, the terminal ends are purely mannose. Very self-explanatory. Okay, now this is the formation of your oligosaccharide. The difference I will note later on. But initially, the formation of the oligosaccharide is through the use of your dolichol phosphate. Now in step 1, you will note that the UDP donates the glucnac to your dolichol phosphate, but not only that, it also donates the phosphate, 1-phosphate. So it releases just uridine monophosphate. But the second step, what is donated is just purely your glucnac. See, the first phosphate is from dolichol phosphate itself, but the second one is from your UDP glucnac. The third step and so forth are just the addition of additional mannose residues in the cytosol. Okay, so steps 1 to 4 occurs in the cytosol. Step 5 and further elongation, what happens here is that a dolichol phosphate will acquire mannose from the cytosol and flip it into the ER lumen. contributing to the elongation of the terminal oligosaccharide chain. And it does this several times until you get purely mannose. And an additional step could be, and again, the dolichol phosphate can acquire glucose from the cytosol and flip it into the cytosol, again, donating it to the lengthening oligosaccharide chain. See? So you have your dolichol phosphate and your oligosaccharide. Now, next slide will show that we mentioned that O-link is post-translational. Here you can see that the N-link is co-translational. So while your RNA is being translated into your protein portion, the dolichol will donate a branched oligosaccharide into your asparagine residue right here. And the oligosaccharide is transferred from dolichol. Okay, I just repeated that. And then the fourth is trimming of the carbohydrate chain, which begins as the protein moves through the rough endoplasmic reticulum. Okay, so take note, everything occurs in the rough endoplasmic reticulum, except for additional trimming and addition of other monosaccharides. This occurs now in your Golgi. So you can see this in your complex and your high mannose. Next. All right. So this is just, this can be found actually in Lippincott. Take note that after those oligosaccharides or your glycoproteins are packed into different or various secretory vesicles. Now, they can be targeted to your life as lysosomes, and these glycoproteins are composed of certain hydrolases, your proteolytic enzymes, and they can be, another secretory vesicle can be incorporated into your cellular membrane and extruded out of the cell, because glycoproteins also serve various functions, right? And most of them are also extracellular. Some of them can also be embedded in the membrane and incorporated as membrane proteins for your cellular receptors. So I mentioned 3, but there are more. Oh, here's another video.
Glycosylation, adding sugars to proteins, begins in the endoplasmic reticulum and continues in the Golgi apparatus. Let's examine the process for one protein. Enzymes in the ER remove glucose and mannose residues from the oligosaccharide chains. Vesicles transport the glycoprotein to the cis-Golgi network, where one of 2 events occurs. Either N-acetylglucosamines attach to the oligosaccharide and are transported to the lysosomes, or enzymes remove more mannose residues. Vesicles transport the modified glycoprotein to the medial Golgi cisternae, where N-acetylglucosamine attaches and more mannose molecules leave the molecule. Vesicles now transport the glycoprotein to the trans-Golgi cisternae. There, galactose and sialic acid join the chain. The glycoprotein is finally ready for transport to the cell's plasma membrane.
Now, this is a summary of your N-glycosylation. Okay, we already discussed that earlier, especially the highlights. Okay, and this is the 3rd time I'm going to repeat this, and never mind, don't repeat this anymore. Same thing, difference between N-linked and O-linked. All right, Carl Joshua Uy, are you here?
Yes, Doc.
Okay, what are the factors that regulate the glycosylation of glycoproteins?
It's I think from what I read, availability of sugars, um, the tissue. I think I think also the amount of dolichol phosphate, like you mentioned earlier, and
I can no longer hear you, Carl.
oh,
Go
hello?
ahead, continue.
Did you hear the first one, or
I
should I
heard the
repeat
first
everything?
two. Go ahead.
Okay. I think I'm not too sure. I only remember those two.
Okay, that's fine. Let's go on L. Hi, L. Can you
Hi,
give the
Doc.
last two?
Wait. Yeah, the last two would be the presence of like the suitable acceptor sites for in the proteins
Again,
and
the presence of what?
suitable acceptor sites in the proteins.
Mm-hmm.
Yes, and the protein transferase. Doc, yes.
Okay. Well, the answers were fine, but to be more specific, because the answers were a little bit vague. Thank you, El. These are the factors regulating glycosylation: availability of sugar nucleotides, not just sugar, because I think what's this Carl said, just availability of sugar. You actually need, the availability of sugar nucleotides. The sugars are attached to the donor. So you're partly correct, Carl. Presence of suitable acceptor sites in the protein, tissue levels of dolichol P, mentioned that, and the activity of that specific oligosaccharide protein transferase. So this is just the generic oligosaccharide protein transferase. Now we don't have to go into the details of each specific enzyme, but just be familiar with these factors. That's all. Those are all in Harper's. All right, Nika Pagador, are you here? Nika?
Yes, Doc.
What is the main defect in eye cell disease?
Eye cell disease. The main defect would be the mannose phosphate 6, Doc.
What happened to that?
They lack of lysosomal enzyme, Doc.
The lack of lysosomal enzyme. Okay, the gist is there, but you can't actually explain it in a biochemical manner. Okay, but thank you for trying. Let's ask.
Thank you, Doc.
Jeswin Jason Sandoval, are you here?
Hello, Doc. Good morning.
Yes. Hi, Jason. What is the main defect in eye cell disease?
So the main defect in eye cell disease is the improper targeting of the lysosomal enzyme. So instead of going inside the lysosome, they're just dispersed in our blood. So there's a problem in the mannose phosphate, which serves as which is involved in the targeting of these enzymes toward the lysosomes, Doc.
Okay, that's good. Thank you, Jason. Now, this is the main defect. You have initially, you're supposed to mention, of course, gene mutation, because everything when you have faulty enzymes, it's either faulty or a gene mutation or a very faulty protein translation. So if you have a gene mutation, you will have a resulting mutant glucnac phosphotransferase. There is a lack of normal transfer of your glucnac1P to specific mannose residues of that enzyme supposed to be destined for the lysosome. But since there is no there's no phosphorylation, there is no mannose 6 phosphate. Mannose 6 phosphate. It's just like an address. When you have mannose 6 phosphate, that correct address will send this enzyme to the lysosome. So without this one, then it's scattered, and then I think Jason mentioned it's going to be extruded out of the cell, and it's not going to perform its function. So what happens when you have proteins which are digested and you have deficient hydrolases or your proteolytic enzymes, so they don't function properly and they are accumulated in the lysosomes, manifesting as inclusion bodies. And then I think in your histo lab, you're going to be able to find various inclusion bodies, more specific than this. But in eye cell disease, yeah, not that certain inclusion body. So again, there is a problem with phosphorylation of your mannose 6 phosphate. So the enzyme, instead of being targeted into the lysosomes for proper functioning, they are targeted elsewhere, sometimes outside of the cell. So in eye cell disease, this is caused by a deficiency of the ability to phosphorylate mannose again. But the characteristics of a patient with this disease have skeletal abnormalities, restricted joint movement, coarse facial features, severe psychomotor impairment, and usually, and sadly, death occurs by 8 years of age. So not very good. All right. So this is just a summary of the O-link and N-link glycoprotein in terms of biosynthesis. So in O-link, the addition of the oligosaccharide is post-translational, whereas in N-link, the addition is co-translational. In O-link, the oligosaccharide chain is synthesized on the protein, but in N-link, it's an N-block transfer of the oligosaccharide chain onto the protein using your tolicol, I mentioned already. But further modification or addition may occur after that. Okay. Enzymes are membrane bound in O-link, but in N-link, not membrane bound. We don't use dolicol for O-link, but your dolicol PP oligosaccharide, you can find that in your N-link. Again, O-link is not inhibited by tunicamycin, but your N-link, the first step is inhibited by tunicamycin. An example of your O-link, I mentioned with very high O-linkages is your mucin. And Vince Edward Pintak. Are you here, Vince?
Yes, Doc. I'm here, Doc.
What's an example of an N-link?
For a while. I'm not so sure, Doc.
Okay. How about Ralph? Ralph, are you here? You're the very
Doc.
jolly Ralph. Hi. You want to laugh
morning.
first before answering? Or give a giggle? Ralph, what is an example of an N-link?
I'm not sure too, Doc.
Hmm.
The one
But you read-
the one that's in the eye cell disease.
The one that's in the eye cell.
I
Because
know.
there was a mannose 6 phosphate, but there was no mention of any N-link there.
Oh,
Hmm.
then I
If
don't.
you-
I'm not
okay.
sure.
But it's okay. It's okay. All right. Maybe I'm going to include that in the exam. Okay. Okay, the third class is your GPI-linked glycoproteins. GPI-linked glycoproteins. So remember, who was that who answered first? Glucosyl phosphatidyl inositol. Glycosyl phosphatidyl inositol. That's the basic composition. Also, it's also called sticky foot. And those proteins I was mentioning earlier, that some examples are, very common ones are your acetylcholinesterase, your alkaline phosphatase. These are all GPI-linked enzymes or proteins. And later on, this will be highlighted, your DAF or your decay accelerating factor found in your red blood cells. So this figure will crop up again. I already explained that earlier. So so this is just the same figure for phosphatidylinositol linked to your cell membrane, your oligosaccharide linked to the protein. Okay, PNH. Brinkley Detisio. Brinkley, hi.
Yes, Doc. Hello.
What's the main problem in the PNH?
Ano, Doc? It will. Wait. There's basically there's hemoglobin in the urine, and it's because there's going to be hematopoietic cells.
Now, no, you explained first biochemically, not symptom-wise. Later na lang the clinical. Because as first-year students, try to focus more on the basic biochemical.
There
It's
would
actually on the
be
slide. So what's the problem in the slide?
there would be a mutation in the PIGA gene, Doc, and
What's
also your
what's
decay.
PIGAG? PIGAG?
Oh, PIGA.
What's PIGA?
I don't know what's the PIGA gene, Doc,
Okay, but what
I know
happens,
about the
especially if you have mutations in the gene? What happens?
your two proteins as mentioned there, your DAF and your CD59, which is your decay accelerating factor, and your CD59 will be affected because they are deficient, Doc, in
They are
paroxysmal.
deficient. So you're saying that the PIGA gene will code for your DAF and CD59? Is that how I understood it?
No, said Doc, but
No. Okay. So what happens?
so there is going to be like lysis in the complement system, and ang
Oh
lysis
no. Before that,
Doc.
I'm talking about mutation in the PIGA gene. Then what happens? PIGA gene will not cause lysis of your complement, lysis by your complement system. Let's ask, but thank you anyway, Brinkley. Let's ask
Thank you, Doc.
Malapitan James. Are you here?
Yes, Doc.
Yeah, what happens if you have mutations in your PIGA gene?
There will be a problem in synthesis of the glucnac, Doc. Because
Glucnac?
I think so, Doc. Because from what I learned, Doc, PIGA would be the phosphatidylinositol na precursor.
And then?
Then if there will be a mutation in the phosphatidylinositol glycan class A, Doc, there will be a problem with the synthesis of the glucnac, Doc.
Of the glucnac? Because you mean the PIGA gene, or you're correct, that is what PIGA stands for, but are you saying that the PIGA will code for glucnac?
The glucnac. I think so, Doc.
Okay. Anyway, sige. But thank you, James.
Yes, Doc.
So here, again, this is also in Harper's, acquired mutation of the PIGA gene of certain hematopoietic cells. You have defective synthesis of your glucamine phosphatidylinositol, linkage of your GPI anchors, because there are certain enzymes which will become defective in the formation of that GPI anchor. Okay, so the enzymes required to form that anchor. Okay, it does not form GLUCNAC. So of course, there'll be decreased amounts in the red blood cell membrane of these GPI anchor proteins. Now your DAF or decay accelerating factor and CD59 is very important because without these proteins, the red blood cells are open to or defenseless against your complement mediated lysis. Therefore, you will have what do you call this? But let me ask. Jan Marie Hilario, are you here?
Yes, Doc.
Why is it termed nocturnal hemoglobinuria?
Because nocturnal, Doc, it usually happens at night.
Okay.
So
But why is it more common at night?
because during at night, Doc, kanang low ang plasma CO2. Wait, Doc.
Low?
There's a drop of koan, Doc, of plasma. Hey, Doc, I'll compose first my thoughts. During at night, Doc, there's increase in carbon dioxide and decrease in pH.
Okay, that's right. You retain more of your carbon dioxide, make your blood more acidic. Okay. And later on, when you go into respite, you'll know actually why. But for now, I think that's enough for you to know. And the hemoglobinuria is because of the lysis of your red blood cells. Very good. Okay. Now, this slide was shown earlier. That's what's not supposed to be there. But Ron David Amatong.
Yes, sir.
Yeah, what's the difference between glycation and glycosylation?
Based on my reading, Doc, glycosylation requires enzymatic activity, whereas glycation it does not require an enzyme. But I'm not exactly sure which enzyme is needed.
Oh no, you don't have to worry about the enzyme, but that's the main difference, and that's good. Okay. Is there a wait? What's happening? Are we? When is there like something happening? Breakout?
Dr. Go, no, just continue, Dr. Go. I'm sorry.
I'm sorry. Okay. Sige. Okay. So we'll discuss now about your advanced glycation end products. That is the last part of your chapter in Harper's. So as you will see that, what is this, by the way? Because we already talked about sugars. Jan, Natalie, what is this structure?
I think it's a glucose, Doc.
Okay, it's glucose. You're right, because there's an aldehyde and it's a 6-carbon sugar. Now your glucose will attach to the amino terminal portion of your protein, and the attachment is, take note, non-enzymatic, forming what you call a shift base right here. So this is the link. And after certain rearrangement, you will form an amino ketone right here, which is termed your advanced glycation end product. We'll discuss more on that in the next slide. Now, advanced or AGEs, we'll term that AGEs, underlie tissue damage in poorly controlled diabetes mellitus. And the glycation of proteins, for example, collagen, which is the most common example in Harper's, a glycation of these proteins causes increased cross-linking and accumulation of increased cross-linking of your collagen. Increased cross-linking of the collagen causes accumulation of plasma proteins. And in the example given in Harper's, LDL is accumulated in your blood vessels, promoting what is called atherogenesis. Okay, Shane, are you here?
Yes, Doc.
What is the role of HbA1c? And what is HbA1c, by the way?
I think, Doc, HbA1c is formed when glycation occurs to hemoglobin
Okay.
A. So if a patient has diabetes, there is an accumulation of HbA1c. So the testing and management of HbA1c levels is used to help patients manage their diabetes.
Okay. Thanks. To elaborate more on that. Oh wait. Okay. So I missed that other slide. It's not here. So you're right. HbA1c is your glycosylated hemoglobin. They can be irreversibly attached to your hemoglobin. And you failed to mention that this can give an estimate of the blood sugar control of the patient for the past, just an average, 3 months. Why do you think, Shane, it's 3 months?
I'm not sure, Doc. I'm sorry.
It's because that's the average lifespan of a red blood cell. Okay. 90 days or about 3 months. So in the next slide, we already mentioned earlier that formula in Harper's, but here it's another figure showing the amino acid and glucose forming. We mentioned the shift base, which occurs from hours to days, and that rearrangement, your Amadori product, occurs from days to weeks, and part of that is your hemoglobin A1c or glycated hemoglobin. And AGE or your advanced glycation end products is occurs or they occur within weeks to months. These are examples of your very unfavorable AGEs. Okay, this can also happen in different ways from your by lipid peroxidation and glucose auto-oxidation. And these are the precursors of your AGEs. Okay, so why is AGEs not good or why are they harmful? It's because in this side of the table, glycated proteins of your extracellular membrane and plasma are extruded, and they form increased cross-linking of collagen. We mentioned that earlier, and they trap LDLs, therefore leading to your atherogenesis. They also damage basement membranes in your kidneys, and they also cause basement membrane capillary thickening, therefore affecting the filtration function of your kidneys. They also activate your NF-κB. Therefore, and we know that NF-κB is pro-inflammatory. So we don't really like too much inflammation. This is exemplified by the increased release of your cytokines and increased procoagulant activity, therefore resulting in endothelial dysfunction. Okay, let's move on to the extracellular matrix. So introduction, introduction. I think you know that already. The functions, you know that, you discussed that in your PBL. 3 major subclasses of biomolecules. You have your structural proteins such as collagen, elastin, and fibrillin-1. And you have your specialized proteins like your fibronectin, laminin, and you have your proteoglycans as well. Okay, so this is just a schematic of how these proteins are formed intracellularly and are extruded out of the cell in your various forms: collagen, elastin, and your proteoglycans. Okay, this is just a mesh of your extracellular components. Let's not go through that anymore. Okay, let's start with collagen. Collagen, you hear that usually in beauty products, aesthetic medicine enhancements, lip injections. But as first year student, I think you should know what the chemistry of collagen is all about. before moving into aesthetics. So introduction. Collagen is the most abundant protein in animals. They constitute 25% of proteins in the human body. It's the main structural protein in most connective tissue, and at least 28 types of collagen are recognized, designated by Roman numerals. But the first, okay, so you have different classes of collagens and corresponding to the different types. For example, fibril forming are types 1, 2, 3, 5, and then 11. And for purposes of discussion, just try to focus on the first four types. Now, type 1 is non-cartilaginous connective tissue. You can find them in bone, tendon, and skin. Type 2, very common in cartilage and the vitreous humor of the eye. And type 3, extensible connective tissue, including skin, lung, and vascular system. Take note, vascular system in type 3. And in type 4, basement membranes. You find basement membrane, an example would be in your renal glomeruli. Okay, let's ask.
Excuse me, Doc.
Oh, yes?
I just wanted to question Doc about the number of collagen. According to Polina, there are 29, but according to Harper's, there are 28. Which one would be best to follow, Doc?
I'm not going to ask actually how many there are, but we're going to, for collagen, anything about collagen, we're going to use Harper's as our text, okay? So don't worry about 28 or 29. That's not going to really come out in the exam. Like, is it 29, 28, 27? No. So don't worry about that. Just focus on the first four, or maybe include the fifth one, okay? But
okay,
just as a guide, anything regarding collagen will be covered by biochem. Okay, but we're not going to go as far as how many types there are, because now that's the existing number, but in the future they're going to discover really more, and that number is going to change. So for information like that, we don't usually ask that in the exam. Okay. Okay. Is Joanna Ortizano here now? You're not absent anymore? Or are you still?
Yes, Doc. Yes, Doc.
Okay.
I'm here.
So you redeem yourself now by describing the primary sequence of collagen. Go ahead, Joanna.
Okay, Doc. Hold on. What do you mean with a primary sequence, Doc? What it's composed of?
Okay, I'll give you a primary amino acid sequence.
It has its collagen molecule is composed of a polypeptide, an intertwining polypeptide chain, intertwining alpha polypeptide chain, and the
primary
alpha
sequence
chains are
is
usually composed of glycine, which is preceded by a hydroxyproline or glycine, and then followed by a proline.
Huh? Is that what Harper says? You just need to tell us the primary sequence. Okay, I'll give you a clue here. Okay. How about okay, thank you, Joanna. Let's leave that for now. I will correct that later. How about Kyla Tan? Kyla?
Excuse me, Doc. Kailatan is not part of the class
I'm
anymore.
sorry. Yeah, I remember. Okay. Catherine Lipat. Are you here?
Yes, Doc. Good morning, Doc.
Morning. Can you describe the alpha helix chain and the triple helix?
For the alpha helix, Doc, it's actually composed of what the student earlier said, Doc, like there's a glycine in the structure, and it's actually followed by either in the there is a structure sequence called lysine XY. So X could either be proline, while for Y is your hydroxyproline. So for the alpha helix chain, doc, it's actually kanang the pro alpha chains that are interspersed that makes the helix chain, doc. Is what I've understood from what I've read, doc.
Okay, thank you, Kath.
Okay, doc.
Okay, you mentioned earlier that the primary amino acid sequence was GlyXY, but you had a confusion as to what X and Y actually are. Okay, so we try to I think you need to read more now and try so that you'll be able to understand that. Okay, and then the alpha. Okay, this is for everybody, not you, Kat, not just you. Okay, and then the triple helix and the collagen molecule. What is the difference between the collagen molecule and the collagen fibril? Okay, so here you have your repeating GlyXY. What is X? What is Y? Okay, I'm sure most of you know. Alpha chain here. Why is it alpha and then not beta? Okay, that was unit 1. We're done with that. And then these alpha chains form a triple helix. Okay, and then the triple helix actually forms the collagen molecule, but the different stages of the molecule we'll discuss later during the biosynthesis. And then finally, you'll have the fibrils or your mature collagen. This is my collagen as seen through an electron microscope. These are the steps in the biosynthesis of collagen. No? So there are steps which occur intracellularly, and there are steps which occur extracellularly. Shira Mercader, are you here? Shira?
Yes, Doc.
Yes. The next question is, what is hydroxylation in collagen biosynthesis?
Is it, Doc, the carbon genosphate oxidized into carbon hydroxyl, Doc?
Can you repeat that?
The carbon hydrogen, the bond of the carbon hydrogen has been oxidized into carbon hydroxyl, Doc.
Carbon hydrogen. Okay. That okay. Do you have a better description?
I think I need to read more, Doc.
Okay. So how about Denise Castilla? Are you here, Denise? You're here, but okay.
Yes, Doc. Good morning.
Morning. What's that? What's hydroxylation?
It's the from what I remember, Doc, but I'm not really sure. Like selected like hydroxylysine residues are glycosylated with your glucose and your galactose.
Okay.
And yes, and then that's when your pro alpha chains assemble, Doc. Yes, the 3 pro alpha chains assemble.
No, not quite there yet. So just stick with hydroxylation. But thank you anyway. Next is the substrate. Jan. Jan from group 2. Hi, Jan.
Hello, Doc. The substrate, I think, from hydroxylation will be the hydroxyproline and the hydroxylysine. Oh, sorry.
I mean, what is being hydroxylated?
Sorry.
So initially, you have your transcription, translation, and then the resultant product is the one that's going to be hydroxylated. What is that resultant product?
Is it the pre-pro collagen, Doc, the one with the polypeptide, the pre the polypeptide chain.
So your answer is pre-pro collagen.
Yes, Doc.
How about Ange?
Doc,
I'll go back to you, Jan, later. Ange, what Jan said pre-pro collagen. Do you have another answer?
the question, Doc, ay what is being hydroxylated, Doc, no? Is it the proline and the lysine, Doc? Lysine residues?
Yeah, but of what? Is it the pre-pro collagen? Is it the pro collagen, or if is there a difference or no? Is it?
I'm not sure, Doc. I'll still read on that.
Okay. So Jan, is that your final answer? Pre-pro?
I think so, Doc.
Okay. Okay. Hydroxylation occurs after the removal of your signal sequence. Removal of the signal sequence will convert your pre-pro collagen into your pro alpha chains, or your pre-pro alpha chains into your pro alpha chains. After the removal of that signal sequence, then there's the time you can do your hydroxylation. So the answer, the substrate, is your pro alpha chain. Okay, not the pre-pro, because it's already been removed, man, the signal sequence. Okay. Location. Nicole and Darius. Are you here, Nicole?
Yes, Doc. Good morning.
Good morning.
The
Where does this all happen?
Initially, the location is the cisternae of the rough ER.
Just the hydroxylation.
Oh,
Kind of correct, but we need a more specific
okay.
location.
Is it the? I'm not sure, Doc. Sorry.
It's fine. It's in the lumen of the RER hydroxylation.
Okay.
Huh? Don't forget that.
Okay,
Okay.
thank you.
Kyla. No, I already called Kyla. Wait, huh? Kevin Lui. Kev.
Yes, Doc. Morning, Doc.
Morning. Is this a post-translational modification or a co-translational modification, this hydroxylation?
I believe it's a post-translational modification, Doc.
What was it again? Post?
I believe it post. Yes, Doc.
Okay. So take note of your answer. You said post-translational modification. I'll get back to you later. Thank you. Next, we're going to call Eden Cornell. Are you here? Kesa?
Yes, Doc.
Yeah. Next is, what are the enzymes involved in hydroxylation?
The hydroxylation is aided by your lysyl hydroxylase and your polyl hydroxylase.
Okay, good. Lilette Ruella. Lilette? You here?
Doc, good morning. I think she's getting vaccinated at the moment, Doc. So
Okay,
maybe
thank you,
she
Jason.
can't answer.
It's fine. Thank you, Jason. Next, Harvey Soros, are you here?
Yes, Doc. Good morning.
Good morning. What are the cofactors involved in the hydroxylation?
The cofactors involved in hydroxylation, Doc, are your ascorbic acid and also your alpha ketoglutarate.
Okay. What else?
Mm,
That's right, by the way. Anything else that you can think of?
I think
if not cofactors, then what else are needed or
the
required?
The
for the
hydrogen,
enzymes to work.
is it not doc, hydrogen bonds? Tama ba?
No.
Essentially, I don't know, doc. I only know those two.
Thank you, Harvey. You need molecular oxygen or O2. And you
Okay.
also need Fe+2. Is that ferric or ferrous?
Ferrous, doc. Fe3
Yeah.
is ferric.
Okay, that's good. So these 4 things, don't forget: vitamin C, molecular oxygen, ferrous, and your alpha ketoglutarate. Yeah, good. Okay. So step 1, we already know transcription, translation, we already know that. And we moved on after the cleavage of your signal sequence to the hydroxylation, which is step 3. And I'm going to emphasize that the hydroxylation, before it's being, this occurs co-translationally. Okay, this is a co-translational modification, the hydroxylation. Then step 4 will be the glycosylation this time of certain, what is that? Glucose and galactose sugars into your growing polypeptide. Then the 5th is 3 pro-alpha chains assemble, and you will have intra-chain and interchain disulfide bonds, which form at the carboxy terminal pro-peptide extension right here. So this is the same thing from the previous slide. And after that, a triple helix is formed and pro-collagen is produced. Pro-collagen, take note, is after hydroxylation and glycosylation. So after those steps, the resultant product or molecule is your pro-collagen. Now, after that, the pro-collagen molecule is secreted from the Golgi into the extracellular matrix, or your ECM. And the next step would be the N-terminal and the C-terminal propeptides are cleaved by pro-collagen peptidases, producing your tropocollagen. So I think at this time, you already know the difference between pre-pro-collagen, pro-collagen, and tropocollagen this time. Remember, tropocollagen after the cleavage of your terminal peptides, your C and your N-terminal propeptide. All right. And then finally, you have your self-assembly of your tropocollagen molecules into fibrils. The fibrils are the mature molecule collagen already this time, formed by subsequent cross-linking to form here mature collagen fibers. Okay. Wait, what is collagen staggered alignment? All right, that's been like a favorite question. But anyway, we'll just go through this very quickly. Ana Luisa Velez. Hi, Anne.
Hi, Doc.
What is collagen staggered alignment or your quarter staggered alignment?
I'm sorry, I'm not really sure.
How about Carlin
I think
Enriquez?
it's like when it's formed into the fibrils, no, Doc. But I'm not really
Yeah,
sure.
but we already mentioned that earlier. How about Carlin Enriquez? Carlin, are you here?
Excuse me, Doc. Carlin
Yeah.
is not anymore with us.
Okay. I need to get an updated list. Noel Madrio, are you here?
Yes, Doc.
Yeah, what's that? Collagen staggered alignment.
Yes, so that's when the the fibrils are basically like located like a quarter away from each other, I think. So there's gaps.
Again.
The
I can't understand that.
the the fibrils are like spaced out a quarter away from each other, 1/4
Further
away.
away. Is that your term? Further away?
No, a quarter, like
Quarter.
one.
Okay.
Yeah,
Sige.
yeah, away from each other, and then so there's gaps, basically in the
Between
the
your
fibrils. Hello? Yeah. And then that's important for the collagen because it allows for tensile strength, I think. And then it also leads to like the bending appearance of the collagen.
Okay. So I'm sure that's what you discussed in your PBL. That's fine. But a more detailed description would be for the alignment. So each tropocollagen molecule displays longitudinally from its adjacent molecule by slightly less than 1/4 of its length. So that's actually the definition. Displaced longitudinally. So you will have your bold lines, if you look at it cross-sectionally, and your lines which have a gap. Now, the ones with the gap, if you look at it through your negative stain, would be the dark bands, whereas the ones without the gap will be your light bands. And the significance, which she mentioned earlier, was okay, on point, so that's fine. Okay, so this is in, okay, so already mentioned that the ones with the gaps are the lightly staining ones. No, sorry, the ones with the gaps, heavy staining in regions with gaps. Okay, so these are the ones with the gaps, and light staining in regions with no gaps. Okay, so this is your collagen fibril. So just to reiterate that one. Okay, so now we move on to the collagenopathies, or your collagen-borne diseases. So you have diseases which come from gene mutation all the way up to here, and you have diseases which are caused by deficiency in the activities of your post-translational enzymes, which you can find here, the last three. Okay, and these are the various collagenopathies. Oh, good. Okay, here. Let's ask Johannes Matthew Manio. Are you here?
Yes, Doc.
What is the cause or defect of
Good morning,
your...
Doc.
Good morning, Ehlers-Danlos syndrome.
Doc, I can't recall, but Doc, I'm sorry.
What about the other questions? Are you familiar? What's the collagen type affected in the classic form?
It is the collagen type 3, Doc.
Classic form?
Oh, the
Okay.
classic form.
It's fine. What about James Malapitan?
Doc, was it type 5, Doc?
It's fine. James Malapitan. Are you here?
Yes, Doc. Yes, Doc.
What's the cause and defect of your EDS?
I'm not really sure about this though. Sorry.
But you discussed that, EDS, in your PBL?
I don't know. We discussed only the one, Doc, the San Felipe. I think we didn't discuss about that, Doc. Pero I just
San Felipe.
Basically, Doc, as far as I
What are your
remember, it is the enzyme deficiency.
polysaccharidosis? What?
It is, as far as I remember, Doc, it is the enzyme deficiency of the proteins involved in the assembly of the primase 1, 3, and 5.
Okay. Anyone else? Let's call Jacob Cabrera. Jacob James, are you here? Is Jacob James absent? Okay. Jacob James Cabrera. How about John Nino de la Torre? Are you here?
Good morning, Doc.
Good morning. What's the
Um,
cause or defect of your EDS?
I try to give my answer on it, Doc. Is it the deficiency of your tenacin X gene?
Are you reading Harper's or you're Googling stuff here? Is that what it says in Harper's?
Kuan, Doc, sa for nag-base I'm basing sa folder 6.1.
What was it again? What did you say?
Then mutation of tenacin X gene.
Okay. So you're supposed to know the cause and the defect by in terms of biochemically, and the collagen type affected in your classic form, and the symptoms we'll try not to focus more because since you're first year, focus more on the cause and the defect. Later na your symptoms more in detail, but it's nice to know lang, and the most serious type. Okay, so your EDS is caused by enzyme deficiencies. Specific enzyme is your lysyl hydroxylase and your pro-collagen proteinase. So the amino acid sequence mutation are found in types 1, 3, and 5. And the most important, just to take note, is that type 4 Ehlers-Danlos syndrome affects type 3 collagen. It's important because type 3 collagen I mentioned earlier is also found in your vascular tissue or your blood vessels. If you have fragile blood vessels, then you are prone to, you're potentially prone to what? Vessel rupture and hemorrhage. And incidentally, you also have soft and stretchy skin. Okay. And your classic, what do you call this? The most common or your classic is your type 5 collagen. Okay. Oh, here's another one. OI. I know you should have really read on this because this is your handout, no? So, Natasha. Hi, Nat. Are you here?
Yes, Doc. Good morning.
What is the cause defect of your OI?
Col1A1 and Col1A2 genes, Doc.
What is the cause and defect?
Ang defect ay sa collagen type
Is
1.
it enzyme? Is it an enzyme problem? Or is it a gene mutation?
Gene mutation.
Okay.
Doc.
So you'll find gene mutation
In the Col1A1 causing defects in the type 1 collagen.
Okay, type 1 collagen. But the mutation will result in abnormal alpha 1 and alpha 2 chains. Diba?
Yes, Doc.
So if you have problems with that, what happens to the sequence?
It gets
The primary
interrupted
sequence. Remember the primary sequence? GlyXY.
by XY.
What happens there?
There, word na yung nag-change sa OSA.
Yeah, you're right. But what is the change?
Oh my God. Bulky replaced by bulky amino acids, Doc.
What is it?
Bulky amino acids, it gets replaced by bulky amino acids.
No, the other way around.
The bulky amino acids replaces the
what?
The amino acids there. The glycine, I think.
Okay, the glycine. Okay, thank you, Nat. All right, so in OI or brittle bone disease, the mutation already mentioned that pro alpha 1 and pro alpha 2 genes of your type 1 collagen. And it's the glycine that is replaced by bulky amino acid. Sometimes you have the thought, but then you just need to switch it around in your head. Okay, so you have abnormal triple helix formation, and usually you have weak bones or brittle bones. which clinically you will find as fractures in infants, kyphotic adults, and poor wound healing. Do you know what kyphosis is? Alport syndrome. I don't know if you read about Alport syndrome, but sige, let's not grill anyone with this one. But you should know the collagen type affected, where is it present in, and the organ system involved. In Alport syndrome, this is an X-linked and autosomal. Type 4 collagen is affected. And we mentioned earlier, you find type 4 collagen in the basement membrane of your renal glomeruli. Okay, so the organ system is the renal or your kidney. Okay, the organ is the kidney. So you'll have hematuria as well as end-stage renal disease as a late sequelae. So this is just mga histologic features. Kani, I'm sure you should have read this one. Agnes Kaingkoy, you here? Agnes?
Yes, Doc.
What is lacking in scurvy?
The reduced activity of dehydrolysis, Doc, your prolyl and your lysine.
In scurvy, you are lacking what?
The enzymes, Doc, the prolyl hydrolases, and
Initially, what causes scurvy?
lack of vitamin C.
Yeah, okay, lack of vitamin C, which someone mentioned earlier is a cofactor. And then, please go on with a more detailed explanation. You're right.
Since this your vitamin C is a cofactor needed for the hydrolases, Doc. It cannot undergo hydroxylation.
It cannot undergo what is it?
The hydroxylation of your proline and your lysine, Doc.
Okay, so what are the enzymes again?
The prolyl and the lysyl hydrolase.
Hydrolase? Again.
The prolyl and
lysyl. You're choppy.
The prolyl and lysyl hydroxylases, Doc.
Okay, hydroxylase. Don't confuse it with hydrolase.
Sorry.
Okay. Sige. So what happens? So what happens now? If these enzymes does not have that vitamin C.
These enzymes are needed for collagen synthesis. So.
Oh yeah, we know because we're talking about collagenopathies. But what is the biochemical defect here? Without vitamin C.
Without vitamin C, there is a without vitamin C, collagen, you cannot synthesize collagen, Doc.
Yeah, you can synthesize collagen, but it's going to be a faulty collagen. But what is the main defect in terms of biochemically, what is the main defect here if you have no vitamin C? Anyway, Agnes, that's good. Thank you. We'll call on... Wala naman. Who else was not called ba? Julianard Marie Deloso. Deloso, are you here? Wala. So duha na ang absent. Jacob Cabrera and Julianard. Or did she quit already?
She's not feeling well today, Doc. I think she already asked permission from Doc Palomo.
All right. How about Shanika Macalos?
Excuse me, Doc. She's currently getting her second dose of vaccine right now, Doc.
So tulun na sila. Okay. So this is the fourth one. The second one with the vaccine and the fourth one. So we'll start again with Abigail Pura. You here?
Yes, Doc.
Oh,
Good
hi.
morning,
Hi,
Doc.
hi, Gail. Good morning. So
Hello,
what
Doc.
happens now? What happens now where Agnes left off?
From what I've read in the book, Doc, you need your vitamin C for the addition of your hydroxyl groups to proline and lysine. So without vitamin C, the hydrogen bonds needed for the final structure of your collagen cannot form. So I think mao na ang defect, Doc.
Yes, you are absolutely correct. There is interchain hydrogen bond impairment. Therefore, you will have an unstable triple helix formation. Okay, thanks, Gail.
You're welcome, Doc.
So in scurvy, vitamin C or ascorbic acid deficiency is the main cause. So you'll have proline and lysyl hydroxylase. And symptom-wise, don't forget those biochemical stuff we mentioned. You'll have capillary fragility, bleeding gums, subcutaneous hemorrhage, and poor wound healing. So these are citrus fruit. Remember, not all citrus fruits are high in vitamin C. They're high in citric acid. That's what's the cause for the sourness of some foods. But like oranges, yeah, they're high in vitamin C. And look at the gums of this guy because of repeated gum bleeding. And here you'll have subcutaneous hemorrhage, but they can also occur smaller, like in a form of petechial hemorrhages. But these are just big subcutaneous hematomas you can find there. Okay. Okay. Menkes disease. Menkes is also important, I guess. Who else? Oh, from my group, isa ang wala na tawag pa. Ay, Angeli. Hi, Ange. Are you here? We
Hi,
called you
Doc.
earlier already. Yeah, unfair because I called you twice. But one mo kung lain makita. What's the cause and basic defect for Menkes disease?
Which
Oh, I'm sorry. It's
Doc?
Angel. Angel. I thought I was calling Angelica. But sige lang, Angel.
Oh, okay.
You didn't have a chance to speak yet. Gel.
Yes.
What's the
Yes,
cause and
Doc.
basic defect in Menkes?
In Menkes disease, Doc, there is a deficiency of copper,
Yes.
which results in defective cross-linking of collagen and elastin.
About the enzyme.
The enzyme is lysyl oxidase, Doc, which is copper dependent.
Okay. So what? No brainer to, diba? Very easy. Okay, so in Menkes disease, there's deficiency in copper. As a cofactor, the enzyme involved is lysyl oxidase, and there is defective cross-linking of collagen. Wait, nano and elastin. Okay, later on there'll be a different one, huh? But anyway, for lysyl oxidase, this is the enzyme. It needs copper. Coprus or coprig? Plus 2.
Cuprous, Doc.
Huh?
Cuprous.
Sure.
Cupric is Cu3+.
Okay. Now, so you'll have defective cross-linking. And then I'm going to explain later on what is the cross-link here between the lysine residue and the alysine residue. Ay, wala naman. Okay. So, since you're the Harvey, what is the linking? What is the link here?
For Menkes, Doc?
No, not Menkes. The normal linkage between the lysine and the alysine residue.
Normal linkage. Not quite sure with the, I only need the defective.
Because you need cross-linking for effective mature collagen. Wala. You're not familiar?
Ang ano ko lang, Doc, is yung mutation sa gene niya. Yun yung ano ko na-remember ko from my pre-med days.
No, not this is your usual or your normal biochemical or your biosynthesis. Forget muna the, what do you call this? The gene defect. just the linkage between collagen chains to form your mature collagen fiber.
Yung covalent bonding between the adjacent fibers, Doc.
Mhm.
Di siya
What is about lysine and allicin?
Carbon to amino, I'm not sure.
Okay, so for now, put at the back of your mind. Let's move on to elastin because there's a different link in elastin. Okay, thank you, Harvey. So elastin, introduction, read that in Harper's. Basic figure. Okay. Now, remember that linkage in collagen. The difference is in the linkage or the cross-linking of your mature elastin fiber. You have 3 allicin and then 1 lysine to form your desmosine cross-link right there. 1, 2, 3, and then 1 over there. Tetrafunctional cross-link. Okay, I'll explain that in the differences between elastin and collagen later. So collagen, you have a relaxed form and your stretched form. Collagen and elastin. Oh, who wants to do that? Well, Sabrina Veralio. Sabrina Veralio. Later naka-Harvey, ha?
Yes,
Sabrina, hi. Can you give maybe 1 or 2 biochemical differences between collagen and elastin?
If I can remember properly, is that your collagen is in a triple helix form, your elastin is kind of just a random coil formation, so that it allows stretching. And also your collagen contains carbohydrates while your elastin doesn't.
Okay.
Also, there's only 1 genetic type for your elastin compared to your many genetic types for your collagen. There's also a presence of hydroxylysine in your collagen and none in your elastin. Sorry, Doc, I can't remember anymore.
It's okay. Okay. Sige. But you were saying something, Harvey? You wanted to add?
I think the elastin, Doc, they are rich actually in proline and glycine. They are poor in hydroxyproline and they lack hydroxylysine as compared to your collagens that they have the proline, glycine, hydroxylysine, and hydroxyproline.
Okay. Sige. You want to change your statement about cuprous and cupric?
The difference between them?
No, you said cupric was +3.
Copper 2, copper 1. Okay.
Okay. So these are the 5 main differences, or actually 7. And then usually in oral exam, we just ask you to name 5. So you just need to be familiar with mostly 5 of them, but then some of them are just repeating lang. So the main difference is that collagen has many different genetic types. Elastin just has 1 genetic type. Collagen has the triple helix, but no triple helix in elastin. Random coil formations permitting stretching. So we saw that in the figure earlier. There is the GlyXY repeating structure, but no repeating structure in elastin. There is hydroxylysine and carbon containing, or contains carbon in collagen, whereas elastin does not. You have intramolecular aldol crosslinks in your collagen, whereas intramolecular desmosine. Remember the 4, the tetra functional crosslink? Yeah. And then the 7th is the presence of extension peptides during biosynthesis, but there's no extension peptides present during the biosynthesis of elastin. Okay, fibrillin and fibronectin. Now, Marfan syndrome is actually it's not very common. And the thing you need to take note here is that in Marfan syndrome, there is a gene mutation on chromosome 15 of the fibrillin 1 gene. This is a large glycoprotein present in elastin-associated microfibrils. Remember, when elastin fibers are extruded out extracellularly, they use the fibrillin as your like a scaffolding so that it can extend and grow there. Okay, so if you have problems with your fibrillin 1, you can also have problems with your elastin. Okay, so in the end, you'll have ectopia lentis, arachnodactyly, and dilatation of ascending aorta, which I'll show in the next figure. So you'll have long, lengthy digits for your fingers and toes. And this is your kyphosis. And you'll have very hyperextensible joints. Ectopia lentis is that the lens is extruded into the anterior part of the eye. You'll have visual problems, of course, and you have dilatation of your ascending aorta. This is the normal aorta, and you'll have here the dilatation because of the weak, what do you call this, vascular wall. Laminin, yeah, let's not do that anymore. Basal lamina. Okay, so let's go to your proteoglycans and your glycosaminoglycans. Okay, now a thing to note here is that they were originally named mucopolysaccharides, and the properties are they're viscous, lubricating, found in mucous secretions. 7 types of glycosaminoglycans. You have your hyaluronic acid, chondroitin sulfate, keratan sulfate 1 and 2, heparin, heparan sulfate, and dermatan sulfate. So these are the 7 types. The functions, you can all find that in Harper's. Some noteworthy is cell migration for hyaluronic acid, compressibility of cartilage in hyaluronic acid, and your chondroitin sulfate. Okay, so some also serve as structural functions. Okay, basic structure of GAG. Kevin Lui again. What is the basic structure of GAG, Kev? Or glycosaminoglycans? Are you still there? Are you still with us, Kevin? Kevin Lui. All right.
Yes, Dr.
Carlo Juntong. Carlo.
Yes, Doc.
What is the basic structure of the GAG?
Your GAG, Doc, is an oligosaccharide. It's a oligosaccharide chain that is attached to a core protein.
Oligosaccharide.
It's a repeating disaccharide Doc, that also has an amino sugar and a uronic acid. I think it's a uronic acid.
Right. Do you want to change your oligosaccharide? You want to change that to something else?
A repeat.
You're right. It's repeating.
It's a polysaccharide, Doc. An unbranched polysaccharide.
It's a very lengthy saccharide, but it's a polysaccharide. So you're right. Thank you, Carlo. Now, the basic structure of the GAG is it's long, unbranched, it's heteropolysaccharide chain. A repeating disaccharide unit is composed of an acidic sugar and an amino sugar as well. Now, the acidic sugar or your acid sugar, these are, of course, modified sugars, and these are also your derived sugar. So the acidic sugar is usually uronic acid, and an amino sugar is your hexosamine. But there are a few exceptions which we will note later. So, okay, maybe Kevin is back from your bathroom break. Are you back, Kevin?
Yes, Doc.
What
Sorry.
are 2 acid sugars in GAG? And what are the 2 acid sugars?
Um, 2 acid sugars and gags. Xylose.
Xylose is not an acid sugar.
Acid sugar.
Sugar acids, I mentioned earlier, I mentioned in the previous unit, sugar acids have the term ending in uronic acid. Uronic acid, remember that?
Oh, are you talking about hyaluronic acid and
Right. And the other one is?
oh no, uronic acid?
What kind?
Glucouronic acid?
Yes. And the other one is?
Idiuronic acid?
Yeah, but here in GAG, more commonly is?
I am unsure, Doc.
Okay, that's fine. At least you got it. Here is the answer. Okay, so uronic acid, you have glucuronic acid and your iuronic acid. The exception is your keratan sulfate, which does not have either of the uronic acids except it has galactose. Okay, so Kevin was right. All right, Ralph, give 2 amino sugars in GAGs. Ralph,
Yes, Doc.
Sige,
I'm
Ralph.
here.
What are the 2?
The amino sugars.
Or hexosamines. You know, these terms can be interchanged. Amino sugars, hexosamines.
Are those the galnak, gloknak, Doc?
Yeah, but try not. What is the word instead of giving the abbreviation?
The N-acetylgalactosamine and the N-acetylglucosamine.
Yes. N-acetylglucosamine or gluknak, N-acetylgalactosamine or galnak. Okay. So in hyaluronic acid, this is the uronic acid, D-glucuronate or glucuronic acid, and N-acetylglucosamine is your amino sugar or your hexosamine. So thing to take note here, hyaluronic acid is very large and it's unsulfated, meaning there's no sulfate attachment to either of the disaccharide repeating units. And it's also not covalently attached to your core protein, as you shall see later on in the figure. So they act as lubricant and shock absorber, and they're found in synovial fluids, vitreous humor of the eye, umbilical cord, and extracellular matrix of loose connective tissue. Chondroitin sulfate, of course, it has sulfate, and the sulfate can be in either position 6 or position 4. It's the most abundant GAG in the body, and they form your proteoglycan aggregates. Heparin and heparan sulfate, main difference here is one is intracellular and one is extracellular. But in heparin, what I mentioned earlier, this is intracellular, 90% of your sugar acid is your iuronic acid. Whereas heparan sulfate is extracellular and the predominant sugar acid is your glucuronic acid. They also have fewer sulfate groups, but more acetyl groups. Dermatan sulfate, this is the structure. And then what's notably distinct about dermatan sulfate is that either acid sugars are present in particular disaccharide units. So you can find both glucuronic and iuronic acid in dermatan sulfate. In KS or keratan sulfate, I mentioned the exception earlier. Instead of uronic acid, you have galactose here. Okay, so you have two types of keratan sulfate, 1 and 2. It's the most heterogeneous of the glycosaminoglycans. Okay, now biosynthesis of GAG is important, but I think the degradation is more important to take note. Okay, so for this, attachment of core proteins, chain elongation, those are all in Harper's. You can read that. And take note here, this is the core protein, right? And these that are attached to the core protein are your chondroitin sulfate, right there, with repeating structures. But before it starts its disaccharide repeating structure, there is an attachment of your xylose to the serine residue of the core protein. Then you'll have 2 galactose residues before those repeating disaccharide units of your chondroitin sulfate. Okay, chain termination, that's the last part, and post-termination modifications. So in post-termination modification, you'll have action of these enzymes, sulfotransferases and apomerases, which can convert glucuronil to your iduronil. Okay, so hyaluronic acid is actually this long polymer, right? And what is attached to them, what is attached to the hyaluronic acid are your core proteins, and what is attached to the core proteins are your glycosaminoglycans, exemplified here as your chondroitin sulfate and your keratan sulfate. Of course, there's also link protein, but that's not very important. And look at this proteoglycan. This is a proteoglycan monomer, by the way, a figure. But if you look at it through electron microscopy, this is what it looks like, the proteoglycans. So this is the main difference between the proteoglycan and the glycosaminoglycans. Wherein the proteoglycan monomer is composed of different glycosaminoglycans attached to a core protein. So in the ECM, you will have your proteoglycan monomers and your collagen fibers and all the other proteins. Okay, so GAG degradation is important because later on, diseases related to problems with degradation can be found. So the glycosaminoglycans, which are found initially extracellularly, are introduced intracellularly by phagocytosis. So once they are inside the cell, they are degraded inside the lysosomes by these certain hydrolases. You have endoglycosidases, exoglycosidases, and sulfatases. Don't worry about that, but just take note that there are hydrolases which degrade these GAGs. So why is it important to know about GAG degradation? I think this is the final lucky Ron Amatong. Hi, Ron. Why is it important to know about GAG degradation?
Because if it's not broken down and it's accumulated, then it could cause certain conditions.
Okay, before jumping into that, what happens if it's not broken down? We already mentioned it's already in the lysosomes. And if it's not broken down, what happens? Before it causes, certain diseases. There'll be what?
Accumulation, Doc?
Okay, accumulation inside the...
That I don't know.
Inside the lysosome.
The lysosome.
Yeah, okay. So it's just, inside the lysosome. Thank you. What is the defect in the mucopolysaccharidosis? Okay, not what do you call this? Not Ron anymore. Leia Gringia. Leia, you here?
Yes, Doc. Morning, Doc.
Good morning. What is the defect in MPS, mucopolysaccharidose?
Deficiencies in the enzyme that degrades the GAGs, Doc.
Correct. Okay. So you'll have mutations in the gene encoding for lysosomal hydrolases. Which are the enzymes responsible? Okay, na Leia, thank you. You can mute
Okay,
yourself
Doc.
now. Which are responsible for the what did it say? Enzymes degrading the GAG. Of course, if there are defective enzymes, you'll have accumulation of those GAGs inside the lysosome, and they are present in various tissues, including the liver, spleen, bone, skin, and the CNS. You need to know this. Someone mentioned about Sanfilippo earlier. So if you really want to discuss about Sanfilippo, then maybe you want to include this N-acetylglucosamine 6 sulfatase in the exam and hexosaminidase A and B. Who was that who said about Sanfilippo earlier?
It was me, Doc, James.
James, do you want to include that Sanfilippo can be converted, can be by
It's very
memorizing all
complicated,
the
Doc.
enzymes? Nah, yeah, I
There
don't
are
think
four
we want to
mga
do
Doc.
that.
Yeah,
Huh?
thank you. Doc. There are
Yeah,
four
let's not
types.
do
Yeah, it's
that
really hard.
anymore. Let's
Yes,
just
Doc.
stick to the first two, hurlers and hunters. No, so before that, mucopolysaccharidosis have, major features because MPS is actually not acute. This is a chronic progressive course, and they affect multiple organs. So you'll have these two organs which are enlarged: the spleen and the hepat. I'm just kidding. The liver. Okay. So these are the symptoms. Just be familiar, but don't memorize all of them, because later on, these are the first two are the ones that actually are the ones that we want to highlight. So you have Hurler's and your Hunter syndrome. The enzyme defect for Hurler's is alpha L-iduronidase, and the enzyme defect for Hunter's is L-iduronate 2 sulfatase. For the symptoms, you just need to know that Hunter syndrome is X-linked. No, X marks the spot for the Hunter, and which symptoms are common in both. Okay, and yeah, let's not do that anymore. All right, good. So we ended just in time. We had a little more time since Dr. Palomo ended earlier. So I guess you're ready now for the exam next week. All right, guys, thank you. And you can have your lunch now. Bye-bye.
Thank you, Doc.
Thank you, Doc.
Thank you, Doc.